Sex pheromones. During a short life cycle of about 22 months, English Channel cuttlefish can perform 4 horizontal migrations from the Normandy coasts to the western part of the English Channel. After a last migration to reach specific mating and spawning coastal areas, cuttlefish aggregate for mating and egg-laying between April and June on the Normandy coasts. This behavior suggests the occurrence of some kind of chemical communication via waterborne molecules that induce the aggregation of mates. Chemical communication in cuttlefish was first demonstrated by Bull and collaborators using Y mazes. They showed that recently laid eggs, ovary extracts and nidamental glands, induced an increase in ventilation rate and the attraction of sexually mature cuttlefish in the arm of the Y maze containing purified extract versus artificial seawater. In the same way, Cummins and collaborators identified a 10-keta protein in Loligo termed Loligo beta microseminoprotein, Loligo beta MSB, that immediately changes the behavior of male squid from calm swimming and schooling to extreme fighting. Loligo beta MSB is synthesized in the accessory sex gland of females, the oviduct gland, the main and accessory nidamental glands, and is secreted with the proteins of the outer tunic of egg capsules. When a male is attracted to the eggs visually, upon touching them and contacting Loligo beta MSB, it immediately escalates into intense physical fighting with any nearby males. Loligo beta MSB was originally discovered in human seminal plasma and prostatic fluids. It is only described in other vertebrates and in the basal chordate amphoxus. It is a highly variable 91 amino acid protein, with 10 spatially conserved cysteine residues that can potentially form 5 intramolecular disulfide bonds, giving resistance to proteolytic cleavage to prolong its activity on the egg surface. In Sepia officinalis, Inault and collaborators identified 3 major related transcripts encoding secreted peptides and expressed in the oviduct gland. RT-PCR and mass spectrometry analyses revealed that transcripts and expression products were co-localized in the oviduct gland. The 2 very similar protein precursors termed SB-alpha and SB-alpha, figure 4, diverge by only 4 amino acids in the alpha-3 and alpha-3 peptides. They yield 7 putative expression products ranging from 1.3 kDa, alpha-5, to 7 kDa, alpha-3 and alpha-3. Amino acid alignments of the 3 protein precursors SB-alpha, alpha and SB-beta. Red asterisks indicate conserved cysteines. Predicted signal sequences are highlighted in yellow, the conserved sequence between SBs in green, differences between SB-alpha and SB-alpha in blue, and potential basic residue cleavage sites in red. All peptides except alpha-1 contained at least 1 cysteine, and 2 of them, alpha-3 and alpha-3, are C-terminally omidated like many bioactive peptides, figure 5a. Schematic diagrams showing the organization of Sepia officinalis pheromone precursors, A, SB-alpha and alpha, B, SB-beta. Precursors encode a complex cocktail of peptides and polypeptides resulting from dibasic cleavages. Black box, signal peptide, vertical black line, potential dibasic residue cleavage site, asterisk, predicted N-linked glycosylation site, S, size residue. The third protein precursor, termed SB-beta, shares 56.7% similarity with SB-alpha and SB-alpha, figure 4, and yields 5 putative expression products ranging from 1.1 kDa, peptide beta 1, to 8.3 kDa, peptide beta 3, with C-terminal omidation, peptide beta 2, disulfide bonds, peptides beta 3, beta 4, beta 5, or in glycosylation, peptide beta 3, figure 5b. For most of the expression products derived from SB-alpha, alpha and SB-beta, predicted post-translational modifications such as C-terminal omidation and disulfide bonds have been confirmed by NLC-MS/MS analysis. These modifications can provide a strong protection against protease and peptidase activity and can be expected to confer the peptides a long life in marine environments. As most of the predicted peptides were recovered by NLC-MS/MS analysis, the processing of SB-alpha, alpha and SB-beta should lead to the release of a cocktail of waterborne pheromones. Peptides alpha-3 and alpha-2 strongly stimulate the contraction of the penis and the gills when they are applied on these parts, figure 6. Bioactivity of synthetic beta-2 and alpha-3 peptides. Beta-2 induced contractions on, A, female gill and, B, penis from a threshold of 10-8 m. No activity on, c. rectum. Alpha-3 induced contractions on d. penis from a threshold of 10-9 m. Therefore, peptides expressed and secreted by a female's accessory sex gland can modulate the activity of a male's genital apparatus. Recent unpublished data show that the protein precursors Sp-alpha and Sp-beta are also able to release a second batch of high molecular weight, 22 to 26 kDa, pheromones secreted with the egg capsule proteins and integrated to the inner layer of the egg capsule. Finally, they are detected in the seawater around it masses once they have crossed the outer layer of the egg capsule. The presence of these high molecular weight pheromones identified by proteomic analysis of the oviduct gland and egg capsule also implies the presence of high molecular weight polypeptides/proteins derived from Sp precursors. These analytical results demonstrate that two modes of cleavage of Sp precursors coexist and generate low molecular weight peptides, prohormone convertase cleavages, and also 22 to 26 kDa polypeptides/proteins released by the eggs into the surrounding medium. The mechanism that leads to the release of both low and high molecular weight pheromones processed from a same protein precursor has to be elucidated. The occurrence of C-terminal omidation for peptides beta-2 and alpha-3 demonstrates that two distinct processings are performed in the Golgi apparatus, which means that low molecular weight pheromones, LNWPs, are not degradation products of high molecular weight pheromones, HMWPs. The present functional hypothesis could be that LNWPs induce mating and the release of oocytes into the mantle cavity, and that HMWPs, as described in Aplysia, facilitate the aggregation of mature cuttlefish in the coastal egg-laying areas. All these data confirm that cuttlefish eggs are a source of pheromones, as described in other mollusks such as marine gastropods of the genus Aplysia. Behavioral tests now have to be performed to clarify the mechanism of action of LNWPs and HMWPs in sexually mature cuttlefish.